All right. So thanks so much. I'm Grant Wells, Director of Innovation and Development. Welcome to today's Maternal and Child Health Research Institute seminar focused on drug discovery for maternal and child health, a small molecule perspective. Today, we're joined by Associate Professor of Pediatrics and Endocrinology, Danny Chou, as well as Ben Bruno, who's Vice President of Clinical Development for Lipocine. And I think what we'll have today is a really nice academia industry collaboration and discussion around differences in pediatric and women's health drug development, particularly focused on small molecules. But before we dig into that, just a few housekeeping items of things coming up and what to expect. First of all, just want to thank our education committee. Here we have a lot of really wonderful people who help put these seminars on and help us review all the content for the year. So just want to give a shout out to each of them. And then a big plug for our largest event annually, which is our 7th annual symposium, which will be on October 24th. You can scan that QR code on the top left to register. That'll happen from 8 a.m. to 4:30 p.m. We have some really great notable speakers focused on information and disinformation in maternal and child health. So we have individuals from Stanford, Michelle Mello and Kurt Langlotz, as well as Sarah Cody, who's our public health director, and Chris Longhurst, who's the CMO and Chief Digital Officer of UC San Diego Health, as well as a number of others. So we hope you at least register for a portion or come for the entire day. Really, really excited about this particular opportunity. And in connection to that, if you do have an abstract that you would like to have featured, the abstracts do not have to focus on information and disinformation, but really can be anything related to maternal and child health. So feel free to submit an abstract if it's relevant to you. We're still accepting those through September 11th, which is coming up quick. But delighted today, really to focus on this seminar. Both Danny and Ben really have a wealth of background that complements this topic really well. Danny specializes in peptide therapeutics as well as those for type 1 diabetes. So I will be interested to kind of hear his perspective on this particular topic. And then Ben Bruno was just recently promoted to Vice President of Clinical Development at Lipocine, which has a really nice balanced portfolio related to men's and women's health. He's not only leading their clinical development portfolio, but also a pharmacist. So from the pediatric and maternal perspective, we'll have a wealth of insights that were really related to dosing and other trends that really are hot topics in this particular space. So without further ado, I'm going to pass it over. So feel free to start the seminar. Thanks, Danny.
All right. Thank you, everyone. For those of you who don't know me, my name is Danny Chou. I have been in Stanford for 4+ years. I am in the endo division of the pediatrics department. And then as you probably see from the second slide of Grant's presentation, so I'm one of the members in the education committee in MCHRI. The reason why we are doing this today is because the chair of the committee, Tony Oro, asked about whether we could do something that's more on the small molecule drug development space for children and pregnant women. And of course, these are the exact area we care about in MCHRI. And then this is actually also a space where, to be honest, there are not lots of ongoing effort going on in biotech and in pharma. And then I, as a member of the education committee, I kind of took the lead, tried to figure out what would be the best way to deliver this seminar, and then really it's a really great pleasure for me to find the company they are Lipocine, and I happen to know Ben pretty well because of our prior interactions when I was still a faculty at U of U, Utah. And today it's really a great pleasure to have Ben to basically talk about the clinical developments in Lipocine, and then their involvement more or less in the children and pregnant woman space. And then to lead to Ben's seminar, I am actually going to talk about some of the challenges and then of the drug, or more on the small molecule drug development in this space. And then basically, I hope to set the tone for Ben's presentation later. So traditionally, there's quite some ethical concerns related to including children and pregnant women in clinical trials. And then this is pretty obvious to think about because there could be potential harm to fetus and then potentially for children because they are still developing their body, and then there could be some long-term developmental risks. So for a long time, there's a pretty delicate balance between how can we protect these populations in the trial while basically for the chance to develop new therapies such that these population can receive effective and safe treatments. And I mean, we are mainly talking about small molecule in this case, but actually, to be honest, normally, even in the case of biologics, one would want to exclude these populations. So let me give you one example that I'm actually very aware of, more related to the development of insulin in our space. So in the 90s, Novo Nordisk, at that time, they were developing a new fast-acting insulin. Back then, they call it insulin X10. So they were doing a phase 1 trial in men, purely for the safety we are talking about here. And then in the meantime, they actually run a longer timeline studies in rats. And then they actually found out that in the rat study they did after 52 weeks, so that's a full year, they actually found out that 30% of the rats develop breast cancer. And then soon, they figure out that there's actually something related to the sex difference, and then the insulin by itself have some sort of mitogenic activity. And then basically they call that pretty early on in a way that they can stop the trial. And then they have some follow-up studies on the men subjects they had in a phase 1 trial. They did not see any long-term problems. And then that's basically one of the potential risks of very early on in including female subjects there. And in this case, it's a good outcome in the end. But the problem is, if we are excluding these populations, that could lead to potentially lack of data in the drug safety and the efficacy for these populations. And most of the time, in the end, in the case of biologics, but a lot of times small molecules as well, in a way that when these drugs are finally FDA approved, they will normally have the specific labeling saying that these are not for children and pregnant women. So for these populations, if they want to use it, the doctors will normally prescribe them off-label, and then potentially with some risk in the limited evidence. And the reason behind it is because in these two populations, there are some specific, physiological differences there. So, for example, we all know that during the pregnancy, there could be, lots of different hormone changes and some other things that could lead to physiological changes. So, for example, the PK and PD of the drug may be very different. And then one of the example would be in second and third trimesters, there could be some very different kidney functions, and that could lead to difference in the nephrotoxicity risk here. For children as well, because they need to undergo rapid growth and some of the developmental changes. And of course, that will affect the drug metabolism as well. One of the key things will be the dosing, right? When we even for something like painkillers for kids, normally they have the specific labels say that within a certain age group, you will have them to have certain volumes, and that's what we know is a pretty safe drug. But for something that potentially can lead to a little bit more side effects, things could be different. Normally, we normalize or try to do some calculations based on the weight, but that may be okay for some drugs, but may not be okay for some other drugs. And of course, for adolescents, things will be even more difficult because of the metabolic changes during the rapid growth area. And off-label use sometimes is required, or you have to get to it, but this is actually pretty difficult. And then potentially because a lot of these drug formulations are designed for adults, not for pediatric population. So even if you try to account for the potential weight or size difference between children and adults, there could still be quite some differences, and then potentially lead to adherent issues. So these are basically what we know so far. Why it has been quite difficult. And then in terms of trying to set up the presentation later, I would like to use a pretty well-known example, and then quite some tragedy, and some of you may be aware of it, but that's kind of revisit that again. And this is the thalidomide case. This is a small molecule drug that was approved in the 50s in West Germany as a sedative drug, and then basically they went through at that time what was viewed as the traditional or the correct way of going through the safety studies. They add some extremely high doses in different animals. They don't see any fatality, and then no obvious side effects, and they believe it's something that can be pretty safe, or they basically describe it as non-toxic. And unfortunately, these actually lead to quite some tragedies when pregnant woman at that time took this drug, it actually lead to more than 10,000 they call it dilated babies, where basically there's quite some significant body part development issues for these babies, and then it actually affected a lot of different countries, and then these later lead to a greater drug regulation and the monitoring. In this particular case, some of you, if you took organic chemistry classes back in college days, some of the lectures may use this as an example, where basically the normal thalidomide molecules are normally taken as a racemic form. So basically that means they have some stereo center there, and it's a one-to-one mixture of the R form and the S form. The R form is the one they know that they can hit the targets, but unfortunately the S form is what I believe is teratogenic. And normally they will talk about that, this is the case where if you can just only use the R form, they would have the drug and talk about the importance of the stereo center. But unfortunately in this case, this is actually not the case, because where the chiral center or the stereo center is, it's actually a place where they got a pretty high acidity there. So basically, they actually rapid interchange between the two forms. So even if some organic chemists can isolate one of the forms, it will actually not be so useful, because when you inject it inside the body, they will still have the form. So basically, in this particular case, it actually means that it's actually the drug testing to be quite important in a way that it's not about what is the specific active molecule, but it's basically the regulation, the drug testing part. You need to basically find a way to exclude the pregnant woman case to avoid the tragedy. This is actually a pretty interesting example in a way that it's actually not really that bad of molecule, because thalidomide back in 1998, so basically nearly 40 years later than its original FDA approval. It was actually approved by, not FDA approval, but approved in West Germany. It was actually approved by by FDA to treat a few forms of different cancers, including its derivative. And then basically back in 1998 was basically used as one of the first line therapy for multiple myeloma. And the studies of this molecule actually increased quite a bit. And there's actually another reason. This actually because now actually we know much more about how these drugs work, because it's actually a small molecule that can engage the E3 ubiquitin ligase complex. And if you remember from your cell biology courses, the ubiquitin molecule, the role of it is to basically to label the specific proteins that our body would like to get rid of, and then basically this is a way to promote the specific protein degradation by the proteasome. And basically depends on the specific feature and the different physiological situation that could lead to their therapeutic effects, like the case of treating multiple myeloma or AML, but in some other cases that could lead to some undesired teratogenic effects to basically to cause problems in limb and the ear development. This is actually a pretty important, I'll say, milestone in the case of the drug regulatory process. In the US, actually, at that time, there's a lot of reports about this, and some of you may know. There's actually because of there's one individual in FDA, she actually saw specific things. So her name is Dr. Frances Kelsey. She actually saw some hints that this drug may be a problem in terms of the development issues. So actually, at that time, FDA never approved the drug, and then that basically keep this particular drug off the market at US, and then potentially avoid the tragedy at US. And then after these events, there's actually quite some reforms in various parts of the world, and then trying to strengthen the regulation. And then they started to discuss all the potential side effects, different testing, and then potentially even more things that's required for the proof of drug efficacy. And also in the woman part, FDA guideline back in after the event, they started to have some of the guidelines in the way that they will exclude women of potential trial-bearing potentials from early trial phases, and then that's basically related to the insulin trial I mentioned here, and then potentially that could be something in the 90s, one of that novel trials may be a problem, but because of this guideline here, that kind of make it still okay, because only men were included in the trial. But they also found out that because of some of these guidelines, I mean, there's a reason to do it, but that also lead to that there's fewer women in the trial, and that could lead to compromise of the research on women health. And then basically now they started to think about how can we include women in trials that we believe that could be beneficial. So I think in this first part of the presentation, I think the take-home message I try to deliver is that the drug discovery process is hard enough by itself. We all know that the percentage of success from the very beginning in the early discovery case all the way to the FDA approval case is low. But this is especially difficult in the space of children and maternal health because of all the potential problems I just talked about, and then some of the prior tragedies that lead to the current situation. So it's actually when you kind of search around the biotech and pharma space, there are not a lot of these sort of activity going on. So it's actually pretty special that today we can have Dr. Ben Bruno to talk about their development in Lipocine more in this space. So with this, Ben, I will give you the podium here to talk about your work.
All right, is the screen showing up okay?
Great.
Thank you.
Yeah, well, thank you to everybody for joining today, and thanks for giving me this opportunity to talk to you about Lipocine and our developing of products for women and women's health issues. As Danny mentioned, we actually worked together at the University of Utah when I was a graduate student and collaborated on a peptide-based therapy product there, so it's nice to talk with you again. And Grant, I'll just mention, he brought up I am a pharmacist, and that is true, but I've worked in clinical development, clinical trials since completing my training. So I don't think you'd want me behind the desk of a pharmacy or on your clinical care team right now. I do have a bit of a specialized area of focus and education. So, as was mentioned, there are definitely some complications and challenges in developing products for women's health, particularly for pregnant women. But I just want to share with you at least Lipocine's point of view, or my point of view, informed by my time at Lipocine, on how you can, in some ways, simplify and streamline this process with the end goal of bringing to market products that can really benefit women and, in some cases, their children as well. So this is our forward-looking statement, just saying this is not, do not invest based on what I have shared with you today. And I'll start by a little bit of background on Lipocine. Really, all of our products are driven by our drug delivery technology. So there's a lot of good therapies out there that work well in cell culture. Some of them work well in animals when you're injecting them, but there are problems when you're trying to develop an oral therapy, even for small molecules, when they are very lipophilic, very fat soluble. The problem is there's an aqueous layer in front of the cells in the GI tract, and these fatty molecules cannot traverse that layer. So Lipocine's technology allows for the oral delivery of these fatty molecules by co-formulating the active drug molecule with these carrier molecules, which effectively hide and bury the drug inside of a micellar liposome, allowing it to traverse the aqueous layer and then deliver it into the systemic circulation. So this allows for a few things. One is improved bioavailability. And these fatty molecules not necessarily have zero absorption, but very low absorption, so we can vastly improve that. But potentially even more importantly is consistent absorption. So with this technology, we can get reliable blood levels of a number of different fatty molecules, regardless of therapeutic case use we're looking at for which specific disease, but we can get these reliable blood levels, which are really a necessity when you're developing a product to treat a condition. A little bit of background on Lipocine itself. It was founded in 1997 by Dr. Bill Hung-Chieh and Mahesh Patel. They're both formulation experts. And at the time, the company really was a formulations contractor. That is, other pharmaceutical companies would come to Lipocine and say, we've got this molecule, it does great in all of our assays, but we can't get it absorbed orally. Can you at Lipocine help us out? And then in the late 2000s, Lipocine began developing products in-house, that is, running our own clinical studies and our own clinical development process. The company actually went public in 2013, and our first drug was approved in 2022. However, Lipocine is a small company. We currently have 17 full-time employees. The breakdown on here is on the right here, showing that about 5 people work in the CMC, that's the actual manufacturing of the tablets or capsules that we will be using in our clinical studies, as well as developing the formulations, running the safety and release testing for these tablets and capsules. And then the clinical development team, which I lead, has 5 team members. And so we oversee the running and designing of the clinical studies, much of the regulatory interactions as well. Intellectual property is 2 people, and they also do a good amount of data analysis, 1 person in quality, and we all work under the vice president of product development all make up the R&D team. We also have a few people on the finance side as well, which you may expect is important and a bit labor intensive, being a publicly traded company. But we have a small group of people working here, although we do have many consultants and contractors that we work with to move these products forward. So now this is a snapshot of Lipocine's current pipeline. You can see that there's a number of different products in it. Today, I'm going to be talking about 3 of them. One is a product for postpartum depression, one for women with epilepsy, and one for prevention of preterm birth. So really, these drugs are looking at the mother's side, not much development in children per se, although, as you'll see, especially the preterm birth project has large implications for children's health. And really, all of these drugs that were being developed are steroid hormones or related to steroid hormones. So they fit into this very fatty, very lipophilic molecules that historically have not been orally deliverable. And once we enabled oral delivery, we really opened up a large number of potential indications to test and run in clinical trials. So you may be asking, you're a 17-person company, your pipeline is about half as many people as you have. So we have 8 or 9 things in the pipeline, 17 people. How does this work? And I think just I'll take a quick aside to the regulatory pathways that are available. And I think these 2 are very well known to people, maybe not the names, but at least in concept. The 505(b)(1) is what's known as a standalone NDA. It's the typical process when a pharmaceutical company comes up with a new chemical entity. They run all the animal safety trials. They run the early phase, mid-stage, late phase, agree to phase 4 trials. But the bottom line is they have generated all the data to support approval themselves. The other end of the spectrum is the abbreviated NDA, or the generic drug process, where there's already a product approved, and all the company is trying to do is show that, look, we can give it and have the same blood levels, the same Cmax and AUC as the approved drug. But there is a middle path here known as the 505(b)(2) pathway, commonly referred to as the repurposing or repositioning pathway. And what's unique about this is that you can submit your NDA where some of the data in the NDA wasn't generated by the drug company. So you can go out and you can look at literature sources, you can look at prior FDA submissions, prior FDA approvals, and really put together a package that has all this information in it, even though the company itself did not generate all of this information. And this is very helpful in that it helps accelerate the path to proof of concept studies. If there's a compound that's been used even 30, 40, 50 years ago, or it sometimes is an endogenous molecule, there's a lot of data on this compound that lets you move fairly quickly from a concept of, we think that this compound will treat this condition, into actually running a phase 2 proof of concept study. However, there are downsides to this, which include the lack of regulatory exclusivity that is afforded to new chemical entities. So typically when you get an NCE, you have a number of years of exclusivity that actually the FDA gives you, regardless of what you have for patents and intellectual protection. But when you do this 505(b)(2) route, it becomes a lot more important to make sure you have patents either on the use of this compound for this condition or the formulation that you're using, in order to make sure that commercially the company is making a sound investment. Also, regulatory protections can be useful in this case as well. There can be certain avenues, things like orphan drug designation, where you can get extra years of exclusivity. Again, really just helping to make it financially worthwhile to try and develop a drug. So as I mentioned, Lipocine really focuses on these steroid-based hormones. So I thought I'd do a quick aside here and talk about endocrinology in the United States. Currently, there's about 8,000 practicing endocrinologists in the US, and compare that to about 60,000 psychiatrists and 50,000 anesthesiologists. There's really a large shortage of both clinician and researcher endocrinologists. If you look at what endocrinologists are doing during their practice days, about half of their patient visits are for diabetes. And interestingly, only about 15% of people with diabetes even see an endocrinologist. So they're spending most of their time on diabetes, but seeing a minority of the patients. Another 20% of their patient visits are for thyroid disorders. And then another 30% are split among oncology, reproductive, neuroendo, or even hypertension and hyperlipidemia. So what that means is that all other conditions have about 1.2% of the clinician visits. So there's over 50 hormones in the body, and about 70% of the time of the clinicians is spent on two, either insulin or diabetes and thyroid disorders. Just saying, in general, the endocrine system is, I think, underappreciated as an underlier of many, many health conditions that we see today. And also, I believe that hormones are attractive as therapeutics due to their pleiotropic actions. So we're used to seeing drugs now, especially small molecules or even some of these antibodies that have very, very high specificity for targeting, one gene or one enzyme or one receptor. But we know that many of these conditions are not going to be solved by targeting one receptor in these very, very complex signaling cascades. So when you have drugs like hormones that can affect multiple systems throughout the body, you may have a better chance of effectively treating some of these more complicated conditions. I think two good examples that are recent approvals that reflect this are Wegovy. It's a GLP-1 receptor agonist. It was originally used for diabetes, but now it's been approved for weight loss. It's been shown to help in end-stage renal disease. It's been shown to reduce cardiovascular risk factor or outcomes. It's been shown to improve fatty liver disease. All of these things are underlying kind of this GLP-1 pathway, but it's interesting that one hormone can have so many effects. And just as another example, a recent drug, Resdifra, that was approved for MASH, fatty liver disease, also known as NASH, is a thyroid receptor agonist. And again, just showing that when you have NASH, which is such a complex and multifactorial conditions, metabolic syndrome, obesity, inflammation, that you can have an agent that acts in a hormone pathway in order to have a clinical benefit. So now moving on to some of Lipocine's drugs that we're developing, first starting with LPCN 1154, which is oral brexanolone for postpartum depression. A little bit on brexanolone itself. It is an endogenous neuroactive steroid. It's also known as allopregnanolone, and it is a metabolite of progesterone. It acts both peripherally and centrally, but in the CNS, it is a positive allosteric modulator of the GABA-A receptor, and it has proven efficacy in the treatment of postpartum depression. So it is approved as a product known as Zulresso, which I'll give some more details coming up. It was extremely efficacious. The problem was it was poorly water soluble. So the delivery was via IV administration, and actually it was a 60-hour IV administration, which really limited its use. And because they couldn't deliver it orally, IV was the only route that was available clinically. And as a metabolite of progesterone, allopregnanolone levels increase throughout pregnancy, reaching a peak right before birth. And after the mother gives birth, there is a precipitous drop in these neurosteroid levels. There's been a lot of research out there that has looked at the association between either the rate of the drop, the amount of the drop, the postpartum levels, and there's always been signals there showing that there's some association with allopregnanolone levels and postpartum depression. However, this had to be studied to see if giving allopregnanolone could help treat the symptoms of postpartum depression. And that's exactly what this product brexanolone found during their development, is that there was robust efficacy with its IV infusion with about 50% remission rates at the end of the infusion. And this remission sustained for up to 30 days, which was the last time they tested for the effects of the drug. And keep in mind, this is a 60-hour IV infusion, which on one side is extremely fast to have a clinical benefit for a postpartum depression disorder. But on the other side, you're having these new mothers come in and have to check into a hospital for 3 overnight stays in order to get this effective drug. And there's also a lot of issues with the IV administration itself. There's a lot of errors and pump malfunctions that lead to higher rates of adverse effects than would be expected without an IV administration. So this is where Lipocine enters the story, and we say, okay, there's this drug, brexanolone. It's highly effective for treating postpartum depression, but nobody is using it because it is so difficult to use with the 60-hour inpatient hospitalization requirement. So we thought, let's see if we can enable the oral bioavailability of brexanolone. And that's when we ran some pilot PK studies, looking really at the formulation screening, some tablets and capsules and different mixtures of excipients to see if we can enable the oral availability. And indeed, we were able to. It was at that point we went to the FDA and said, there is this drug approved, Zulresso. Everyone agrees it's safe and efficacious. However, the IV administration limits its use. So what if, instead of doing our own clinical development path in terms of efficacy studies, instead of running trials in women with PPD, what if we showed we had similar blood levels as the IV product, but we delivered it orally? And at that time, FDA agreed at a high level that that seemed reasonable. And so from there, we went on to some dose finding studies, both single dose and multi-dose, even running some crossover studies with the IV product to see if we can get similar blood levels. And once we had the results of those studies, we went back to the FDA, shared our data, and we came to an agreement on what the precise approvable endpoints would be to show that, yes, your oral administration has similar exposure than the IV and therefore is expected to be similarly efficacious. And I just wanted to call this out. I think it's something that may not be appreciated in terms of pivotal studies, is that when you're running a phase 3 study, you're really using the exact same tablets that will be used in a commercial sale. So that is, whatever is going to be sold to the public if the drug is approved, those lots have to be used in your pivotal study. So what we did then is, once we identified our dose, we scaled up our commercial lots. We ran a small study just to make sure that everything was reacting the same way, given that we were no longer manufacturing it at Lipocine, but at a commercial manufacturer. So then we submitted our pivotal study to the FDA, where once again, they have a chance to give comments on the protocol and the endpoints. And then we proceeded to our pivotal PK bridge study. And in this study, women came in for 2 visits. At one visit, they got the IV drug, and in one visit, they got the oral drug. And what we found was that there were indeed similar blood levels with the 2 treatments. Both Cmax and AUC, which are the key bioequivalence criteria, were very similar between the 2 treatments. And given that we had an oral treatment that had some more peaks and troughs compared to an IV infusion, FDA also asked us to look at a parameter C trough, which also we met the criteria that we agreed upon with the FDA. So this was all, a relatively fast process. We submitted our IND in 2021 and ran our first studies there. Now we're through our pivotal PK bridge study here in just a number of years. So just showing kind of the power of the 505(b)(2) pathway to really accelerate the development of products. And in all of our studies, this product has been very well tolerated with minimal adverse effects. So running the pivotal PK study wasn't the end of it. There's also labeling studies that need to be done. These are very ubiquitous throughout drug development, that basically every oral product being developed needs to do a food effect study to see what happens if the drug is taken with food versus without food, and also evaluating the metabolites that are formed after the drug is administered, just to see if there's any active metabolites, what are the level of those metabolites. Are there things that then we have to worry about in terms of drug-drug interactions or potential adverse effects if you have buildup of these metabolites? And now we're looking to submit our NDA for this product in Q4 2024, so just at the end of this year. So it was about 3 years total from concept through NDA submission. And I should say, we haven't submitted yet. Our target is the end of the year. So I just wanted to show real quickly, some of you on the call may have heard of Zurzuvae or zuranolone. It was actually developed by the same people who developed the IV product because they couldn't do it orally. So what they did is they made a synthetic derivative of a natural hormone just to help get it absorbed in the GI tract. And that led to some differences in the efficacy of the drug, and it wasn't quite as quick into efficacy in that it took 2 weeks to treat the postpartum depression versus about 2.5 days for the IV treatment. And there were some other concerns that came up in terms of drug-drug interactions, as well as potential risks of this synthetic molecule on the infant as well as on the milk production of the mother. That's one benefit of working with these endogenous compounds, is that the baby is basically bathing in this in utero, given the high level of progesterone in its metabolites. So there's a lower level of safety concerns with the exposure of the newborn baby to this compound. So moving on quickly to LPCN 2101, which is a drug we are developing for women with epilepsy. We think there are some specific concerns in this population. It's actually quite a large population. About 900,000 women of childbearing age suffer from active epilepsy in the United States alone. And one really surprising fact to me was that about 80% of women with epilepsy report at least one unintended pregnancy throughout their lifetime. And that may be due partially due to the drug-drug interactions with the anti-seizure medications and contraceptives, in that the ASMs can actually decrease the blood levels of the oral contraceptives. It can lead to unintended pregnancies. And especially in women with epilepsy, that can be a serious concern because so many of these anti-seizure medications have fetal toxicity risk. There's other unmet needs in this population as well. Just overall epileptic patients, there's about a third of them that are not responding despite trying multiple therapies. And also there's mood disorders that are comorbid with epilepsy, things like depression and anxiety. And also there's drug-drug interaction risks, not just with the anti-seizure medications, but also with just other medications like birth controls that these women are taking. So what we are looking to do is develop a therapy with that gives good seizure control with low or no teratogenic risks, and looking to bring a new mechanism of action into the epilepsy drug portfolio, and also to potentially address these comorbid mood disorders. So a little about LPCN 2101. It is a positive allosteric modulator of the GABA receptor. Unfortunately, I can't share too many details on this. I will say it is a different molecule than the one we just discussed in allopregnanolone. And it is, again, an oral dosage form that allows for the administration of this agent. And back to this 505(b)(2) pathway, this compound has been studied in the literature. There are multiple epilepsy animal models that demonstrate the efficacy of the molecule in a wide variety of types of epilepsies. And also in humans, in women that are pregnant, the low levels of this compound predict worsening of epilepsy. So typically during pregnancy, progesterone levels increase throughout pregnancy. However, if this particular molecule does not increase throughout pregnancy, women are actually more likely to have worsening of epilepsy. So in terms of our clinical development path, given that it is an endogenous molecule, given it had been studied in animals before, we really ran just an animal toxicity study, and then we're able to submit an IND and get that opened with the regulatory body in order to start the human clinical trials. And to date, we've administered about 84 doses to 37 participants across our 4 clinical studies. The drug was well tolerated. And importantly, there was a dose linear increase in exposure, so that once we identify what is the effective dose in humans, that you can then basically choose the strength of the formulation to reach that therapeutic threshold. And in terms of next steps, we are looking to run a proof of concept study. There's a condition known as photoparoxysmal response epilepsy or photosensitive epilepsy, where it's a very good screening model in order to test for the anti-seizure effects of a given medication, where you can get relatively quick readouts of the effects of the drug, even throughout just a few days of the clinical trial for each individual participant. And importantly to it, it can help you aid in dose selection for future studies. I will say that this drug is not a focus of interest right now of active clinical development for Lipocine, just due to the prioritization of our different projects in our pipeline. However, we are always interested in talking to clinicians who may be interested in looking at this agent or learning more, I'd be happy to discuss if there's any interest on the Stanford side. And one last one in our women's health portfolio is LPCN 1107, hydroxyprogesterone caparate for preterm birth. Of course, preterm birth is a major and growing problem in the United States. Our rates are embarrassingly high compared to not only the developed world, but even parts of the developing world. And this problem is actually getting worse, not better. In general, the underrepresented minorities are even having higher rates than the rest of the United States. And of course, the earlier the preterm birth, the higher the risk to the newborn infant. And it's actually a leading cause of death in newborn babies. There actually was a product approved, which some of you are likely aware, an injectable product known as McKenna. It was approved to reduce the risk of recurrent preterm birth. This molecule is shown here on the right, which you can see it's another steroid-based compound. It is very fatty, very low water solubility. So it was delivered via a weekly injection. And to date, there is no oral therapy. I also wanted to just walk through this McKenna, this product that was delivered, because I think it's an interesting representation of how work that comes out of academic institutions can lead to a drug being approved, even if there are definitely some hiccups and some concerns with this in terms of the availability of the product. So the trial that first showed there was efficacy in the prevention of preterm birth was run through the NICHD and the Maternal Fetal Medicine Unit, led by Paul Meese, and published in 2003, showing that there was a large reduction in preterm birth with the use of injectable HPC. And then a few years later, actually, a pharmaceutical company licensed this data and submitted it for an NDA approval. The NDA generally voted yes, that there was efficacy of the product, and they did not actually give approval at that time. But in the meantime, ACOG, American College of Gynecologists, updated their guidance documents recommending this therapy. And the problem was that there was a single study, multiple trial sites, but a single study that showed it prevented preterm birth and that it reduced the number of babies born before 37 weeks, but it did not show the clinical benefits to the babies. So FDA asked that a post-approval study be run, and it was actually initiated before FDA would grant the approval. And even when the FDA did grant that approval, they gave it an accelerated approval, which is a contingent approval based on the confirmatory study showing positive results. So the drug was approved in 2011, and this the trial was not completed until 2019. So, almost 7 years after the approval was given. And unfortunately, the results of that study were negative, which led to a lot of discussion as to why the results were negative, as well as concerns from clinicians who thought they would have nothing to treat, to give women who are at risk for preterm birth if this drug was removed from the market. However, in early 2023, the drug was eventually removed from the market due to lack of efficacy. FDA did not have concerns about safety of the drug, but rather they said that since there is no efficacy, the risk to benefit ratio is all risk. So now you may be asking, okay, this drug failed its pivotal trial. Why does Lipocine think that they can succeed with this same molecule? And unfortunately, I cannot share that rationale with you today. However, I will say that we have strong support from the Maternal and Fetal Medicine Unit out of the NICHD, as well as Drs. Tracy Manick and Dr. George Sade. In terms of our clinical development, we have run numerous animal studies in terms of toxicity, formulation screening, as well as dose finding. So these were, of course, all done before the compound oral formulation went into humans. And since then, we have run numerous studies in both healthy as well as pregnant females. We also have some regulatory protection here in terms of orphan drug designation as well as patent protections. Again, I mentioned that because it is of a particular interest to a pharmaceutical company when we're developing a drug that is not a new chemical entity in that it has been used and studied many times before. Then we have a phase 3 protocol basically sitting on a shelf after it's been reviewed by FDA and key opinion leaders and been signed off on. However, we're looking for potential partners to help fund and run this study, given that we are a smaller company and this trial would probably take 5 years to run or so. We're looking for some support either from government institutions like the NICHD or other pharmaceutical companies. So I was going to talk briefly on our men's health product just to show that, we're a bit funny that we have products for women's health and products for men's health, and actually some products for both men and women, but just showing that these steroid-based hormones can be effective in a number of conditions. I'll just skip through these, showing that we had really positive beneficial effects in NASH, or non-alcoholic fatty liver disease. And also, we just completed a trial in men with liver cirrhosis on the liver transplant list, where again, we showed that there were improvements in muscle mass, which led to improvements in clinical events like hepatic encephalopathy. And today I won't talk about these two other products, LPCN 2401 and 2203, but again, those fit in the pipeline as small molecules, but very lipophilic steroid-based molecules that we think have high potential to treat these conditions. So in conclusion, about hormones themselves, I believe they are potent and effective pleiotropic molecules that may become mainstay treatments for many conditions. However, oral delivery will be a constant challenge. In terms of developing products for higher risk populations, including maybe pregnant women or children, I think that this could be actually a very interesting area to look, given that there's a wealth of knowledge on these compounds in terms of their safety. So it may assuage some of the concerns or help address some of the risks that can be faced with novel chemical entities that have not been tested at all in these populations. Also, the 505(b)(2) pathway allows for the accelerated clinical development. Something to keep in mind that maybe these are not quote unquote sexy molecules, and that they aren't new chemical entities. They don't have great binding values or great mechanism studies or great developmental stories, but they can be very effective treatments, and they can, by going through this pathway, you can really accelerate clinical development. And it's important to note that once you develop, one formulation of these compounds, there's really a wide range of conditions that could be treated with these types of therapies, including for neuroactive steroids. There's interest in not only epilepsies, but essential tremor, pain, and other CNS disorders as well. And we are always looking to collaborate with investigators at academic institutions for investigator-initiated trials. In fact, we have recently started some of these conversations in a couple of our projects, really looking for these academics actually reached out to us and we're like, we have seen your results, you presented at conferences. We are interested in scientific question X, and we'll work with the investigators, see what we can provide, not only the drug, but also any kind of other support for the clinical studies. So if there is interest in any of these things, I'd be very interested to meet with you. With this 505(b)(2) pathway, you are a bit of a jack of all trades and master of none, in that you're really getting to work on a number of disease states, but it's not like I get to spend all of my time in one of these areas, which really makes it important to build relationships with the experts in the field to support the development. There's only so much you can learn in reading the literature and the textbooks. Talking with the experts is so key to the development of these drugs. So with that, I'll just say thank you again for your time and your attention. This is just a picture of Salt Lake. It's a beautiful city, and I recommend coming and visit. Thank you all.
Ben, Danny, that was absolutely wonderful. Thank you so much for your wonderful seminar today. We can now open it up for questions. We've probably got a few minutes left, so feel free to either come off mute or use the chat, and I can kind of moderate. Tony, do you want to go first?
Oh, I'd love to. Thanks so much. Ben, Danny, amazing job. Really appreciate it, especially the different pathways for FDA. It sounds like you're using endogenous molecules for the 505(b)(2) pathway, which for steroids. Just for the audience, is there other 505(b)(2) pathways, not that Lipocine does, but that other examples of besides hormone therapies that might be useful? And also, have you ever thought about the generic pathway or some other types of pathway, the new pathway that would be helpful? I just want to give the audience a little bit more a broader view.
Sure. Yeah, good question. So we mostly focus on these endogenous molecules. I will say this preterm birth product is not an endogenous molecule, but the 505(b)(2) pathway is not just for endogenous compounds. It's really for anything that has any data that you want to use that you didn't generate yourself. So if there's, it's amazing. You go back and look at literature even from the 60s, 70s, 80s, especially in some of our androgen-based therapies. We'll use some of that specifically, maybe not for the approval package, but when you're going to the FDA and even trying to get an IND opened, it can really help to speed that process along. And also, it can help on the back end as well in terms of streamlining the development. They don't have to be endogenous molecules, just any sort of clinical data that you have access to.
Thanks so much. Any other questions? I don't have any in the chat. Anyone off mute?
Yes, I have a couple of questions. Can you hear me?
Yes.
Yes.
Okay. Thank you for that very informative and interesting lecture. I have two questions. The first one is regarding the delivery molecule. You mentioned to increase the oral absorption through the GI tract. To me, sounds like that delivery molecule has anti-pathic property. So I'm wondering whether, in fact, the outer part, the hydrophilic portion, can potentially interact with endogenous molecules like the bile acids, bile salts, which also has that. And the second question has to do with your anti-seizure investigated molecule. You mentioned that it's being a allosteric modulator of the GABA receptor. And if I understand correctly, a lot of the anti-seizure medicines, they are also going through that pathway. So is there potential drug-drug interaction there?
I'll start with the second one first, that yes, there are a number of drugs that work on the GABA system, either directly or indirectly. The compound itself does have actually a unique binding site from anything else that is known in either, the benzodiazepines or the barbiturates or even alcohol on that system. However, that being said, there can be overlapping toxicity or adverse effects where if you're taking another CNS depressant, it can be made worse by taking this similar class of drug. In terms of the interactions with bile acids, this is not something we have seen clinically. We've run some human ADME study too, looking at hepatic recirculation, basically going back into the GI tract, and there was very minimal of that, which you would expect to see if there was a strong interaction with the bile acids.
Thank you. Appreciate your answer.
I have a quick follow-up. Just so it sounds like for drugs, also for metabolism, do you just in terms of where research should go in the future for this area, do you think understanding fetal metabolism would help us to then reuse drugs better? Or like, where would you put your academic research if you had, like, let's say we gave you $1 million in an academic setting, where would you invest it to make this happen faster?
It's a good question. I honestly haven't really thought about it from the basic research, earlier development side of things. I just love it when everyone publishes papers and I get to read them after the fact. I do think it would be very good to look more at the differences in endocrine systems in kind of, even from newborn, that's a different than even 1 year, 10 years old, and, pre-puberty timeframes. There's a large differences throughout that timeframes. And I think it'd be interesting to see the associations between certain disorders and underlying endocrinopathies or endocrine changes. I'm not sure exactly how I would design that. It's a good question to think about, though.
Thanks.
Hello, can you hear me? Hello?
Yes, go ahead.
Yes, sorry. So thank you very much for the presentation. I just wanted, like, like you pointed out, like the molecules we share highly, like steroids and highly lipophilic, and so mostly like delivery, oral delivery is going to be a challenge. While I believe like for the developed world, this is not going to be a problem because at any point in time you have clinicians who can deliver it appropriately. I don't think it's going to be the same, especially for the developed world. If you most of these drugs are going down line, I know excluding significant population of the world from some of the drug solutions in terms of this. I think most of them like intravenously delivered. Is there any chance like we can most of these can in terms of delivery make them oral to not exclude the rest of the people who have challenges to access to clinicians who can proper injections in those remote areas and all.
I'm sorry, I'm not sure if someone else was able to.
I
hear
think
that.
I was kind of waiting. Mohammed, maybe you could chat that. There was a bit of an echo in your microphone. It made it really difficult to hear.
Hello?
Yes.
Is it better?
Yeah, I think that echo is still there. Maybe you could use the chat and just chat your question and I can ask it for you. Anyone else while we're waiting for that?
Well, maybe I'll just add one more thing that I would recommend if even someone's running an investigator-initiated trial. I really have found the FDA to be very communicative, maybe more written responses nowadays, but just even having a pre-IND meeting with them, sending them questions, asking how certain things would be received from them can really be a good use of your time and resources to just try and get to whatever your burning questions are out to the FDA to see if they would agree with you. And like I said, it takes them about 60 days to get back to you, but it can be a very, very informative step to take.
Thanks so much, Ben. Let's see. Still haven't seen that question.
We can also just email it to Ben as well.
Yeah. Sure. Yes, and there will be a recording posted on our website. So for those that were joining later or wanted to review again, you can find that in the information on our website. And then for those that weren't able to answer the questions, we can answer those later, either by email or if you think of anything else, feel free to reach out to us. So thanks so much for your time. And Danny, Ben, really a pleasure. Thanks for this wonderful overview and really a great seminar for an hour. We got a ton of really wonderful information. So I can't thank you both enough and hope to chat with you soon. Bye, everybody.
Thank you, Grant. Thank you,
Thank you,
everyone.
Ben.
Bye.